In this segment, I'm going to talk about the renin angiotensin aldosterone system. The liver secretes a precursor to angiotensin called angiotensinogen. The OGEN suffix indicates a precursor. The kidney then secretes a substance called renin. Renin converts angiotensinogen to angiotensin 1, which is only weakly effective. The lung then produces a substance called angiotensin-converting enzyme, ACE or ACE. Although the lung is the primary source of angiotensin-converting enzyme, other endothelial cells are known to produce ACE. Renal arterioles, for example, are a source of angiotensin-converting enzyme. ACE converts angiotensin 1 to angiotensin 2. Angiotensin 2 triggers the adrenal cortex to secrete aldosterone. And it's the job of aldosterone to signal to the kidney to reabsorb sodium and therefore water from the distal convoluted tubule and cortical portion of the collecting duct. Aldosterone is secreted in response to increased levels of plasma angiotensin 2, also to an increased concentration of plasma potassium, and increased plasma ACTH concentration, which is adrenocorticotropic hormone. ACTH is not directly involved in sodium maintenance. ACTH is released when plasma steroid concentrations are low, and that includes aldosterone and cortisol. Well, in this crude picture of the glomerulus, we've got glomerular capillaries here, an afferent arteriole leading in and an efferent arteriole leading away. This is the distal tubule here, and along this edge of the distal tubule we see Macula Densa cells. These are the cells that are responsible for sensing sodium chloride in the fluid. Right next to them we see these granular cells that contain packets of renin. Lining the afferent and efferent arterioles, there are smooth muscle cells. So we're going to talk about the control of blood flow to the glomerulus and the control of renin secretion. The macula densa is positioned to monitor sodium chloride through this sodium-potassium 2-chloride cotransporter, NKCC2. The 2 means type 2. Type 2 transporters are located on the apical membranes in the tubules. The NKCC1 is located in other membranes, including the basolateral membrane of the pancreas. A high sodium chloride entry into the cell indicates high GFR. The fluid is moving through the tubules too fast to be reabsorbed. As a result, too much sodium and chloride is appearing at this NKCC2 transporter. As sodium and chloride rise inside the cell, water follows, the cell swells. Swelling of these macula densa cells causes the release of adenosine and ATP, both of which cause an increase in calcium concentration in the sarcoplasm. A rise in calcium concentration causes smooth muscle contraction. This restricts flow through the afferent arteriole, thereby reducing GFR, reducing tubular flow, and reducing sodium chloride delivery to the macula densa. In addition to the influence on smooth muscle cells, the macula densa also influences the granular cells. Under high sodium chloride entry, the cell once again swells and releases adenosine. Adenosine in this instance triggers an inhibitory G protein, which then inhibits the production of cyclic AMP, and cyclic AMP is produced either through norepinephrine from the autonomic nervous system or from a prostaglandin-triggered cyclic AMP. Cyclic AMP causes the phosphorylation of protein kinase A, which then causes the translocation of renin-secreting vesicles to the membrane where they fuse and are released. And I will always point out that ANP, atrial natriuretic peptide, acts to inhibit this pathway through cyclic GMP-mediated protein kinase G, which inhibits this translocation process. So under high sodium chloride entry then, renin secretion is inhibited. So under conditions of low sodium chloride entry, these cells begin to shrink. The shrinkage of these cells turns off the adenosine release, and the low chloride concentration triggers a p38 MAP kinase. This then triggers the transcription and translation of cyclooxygenase 2, which in turn is converted into prostaglandin E2, which is then released, triggering the production of cyclic AMP. Protein kinase A then triggers the movement of these vesicles to the membrane, and renin is released. So under low chloride entry, we see an increased renin secretion. Under high chloride entry, decreased renin secretion. As I mentioned in one of the earlier slides, aldosterone secretion is stimulated by angiotensin 2 and by high potassium concentration. angiotensin II through the second messenger system causes a release of IP3, which triggers calcium release, which then triggers this CREB protein. In addition, diacylglycerol, DAG, triggers protein kinase C, protein kinase D, and that enhances this production of CREB. This then causes the transcription and translation of aldosterone synthase. Now, aldosterone synthase, we can jump all the way down to here, is responsible for converting corticosterone to aldosterone, which will then be released. Under high potassium concentration, membrane potential rises. This causes voltage-gated calcium channels to open. Calcium entry into the cell eventually triggers this same process. Aldosterone enters the distal tubule and the cortical portion of the collecting duct, binds to a mineralocorticoid receptor, enters the nucleus, and promotes the production of more epithelial sodium channels, more sodium-potassium ATPase pumps, more renal outer medullary potassium channels, and proteins which activate these pumps and these channels. Aldosterone promotes sodium uptake in these channels, which is then pumped out through this pathway. Water follows, so sodium and water retention occur during aldosterone stimulation. In addition, potassium channels increase on the apical membrane. As sodium enters, the positive charge forces another positive charge, that being potassium, out of the cell. Aldosterone causes potassium excretion and sodium retention. A decrease in plasma volume will increase renal sympathetic nerves. This is a pressor response. A decrease in arterial pressure will have a direct effect on plasma renin, and plasma volume and arterial pressure going down causes a decrease in GFR. That causes a reduction in the flow to the macula densa, a reduction in sodium chloride concentration in the macula densa. Cells shrink, causing plasma renin release. The renal sympathetic nerves through norepinephrine secretion causes renin release, and there's a direct effect on decreased arterial pressure, also causing plasma renin release. Well, renin, through a couple of steps, promotes an increase in plasma angiotensin II. Plasma angiotensin II then stimulates vasoconstriction and an increase in thirst. Vasoconstriction will help to raise blood pressure, an increase in thirst will help to raise plasma volume and therefore pressure. Angiotensin II also increases aldosterone secretion, which increases sodium and water reabsorption and decreases sodium and water excretion. The goal here then is to raise plasma volume and raise arterial pressure. Angiotensin II's effects then occur on arterioles to increase vasoconstriction. Angiotensin II works on the cardiovascular control center to increase cardiac output. It also works on the hypothalamus to increase vasopressin, and vasopressin is another name for antidiuretic hormone. We'll talk about antidiuretic hormone in another section. And it also triggers an increase in our thirst centers. Plasma angiotensin II also stimulates the adrenal cortex to release aldosterone, causing sodium and water reabsorption. Antidiuretic hormone, an increase in thirst, and sodium and water retention work to increase plasma volume. Vasoconstriction and cardiac output work to increase blood pressure. An increase in plasma volume also then leads to an increase in blood pressure. Hypoaldosteronism leads to hyponatremia and hyperkalemia. Too much aldosterone leads to too much sodium reabsorption and too much potassium loss. Impaired aldosterone receptors, or epithelial sodium channels, leads to the same condition, but we call it pseudo-hypoaldosteronism. If the aldosterone receptors aren't functioning, then angiotensin II will not stimulate aldosterone release, and if the epithelial sodium channels are malfunctioning, then sodium will not be taken up in large quantities in this portion of the kidney. Hyperaldosteronism, which could be the result of adrenal tumor or pituitary tumor causing the release of too much ACTH. This will lead to hypernatremia, too much sodium uptake, and hypokalemia, too much potassium loss, which then leads to hypervolemia, hypertension, and edema. Little syndrome is a mutation that causes the epithelial sodium channel activation to be quite high, so we're taking too much sodium and water into the cells and into the plasma, leading to hypokalemia, and this condition also results in hypervolemia, too much plasma volume, sodium retention, and edema. In the early stages of congestive heart failure, we may see some reasonable compensation. So if the disease is not severe, renin secretion will increase, and this will restore hemodynamics. So increased plasma volume because of increased renin angiotensin aldosterone will cause an increase in sodium and water retention. This will cause an increase in the venous return and an increase in cardiac output from the Frank-Starling mechanism, which I discussed in an earlier section of the cardiovascular system. In this new state, we may actually see systemic arterial pressure normal, renin activity and aldosterone concentrations coming back to baseline. So it's very difficult to see that there's actually anything wrong here. However, there's a price to be paid, and that price is the persistent volume expansion of the plasma will cause increased ventricular wall tension because of the higher venous return and edema because of the higher venous pressure backing up to the tissues or to the lung. So there is a price to be paid for compensated heart failure. As the disease progresses, the system becomes decompensated or uncompensated, and now we begin to see some serious consequences. That concludes this talk on the renin angiotensin aldosterone system.